Am I audible to everyone? And your answer yes or no?
Yes, sir.
Yes, sir.
Okay, good. So, okay, fine. So in the previous class, we have already seen about the recombinant DNA technology. Okay, so in the recombinant DNA technology, we are talking about the different tools. Okay, so as we know that if we are doing the recombinant DNA technology, we need some tools. We need some tools, and these tools will be comprises as same which you are seeing in the images. Okay, means you need scissor. Okay, you need something to make a clones. Okay, so in the previous class we have already seen that as a scissor, okay, the molecule or the enzyme which is working as the scissor in the RDT, that is the recombinant DNA technology, is nothing but this. Okay, like this structure, and these are called as the molecular scissors. Molecular scissors. Okay, and the molecular scissors in the recombinant technology or the biotechnology are particularly called as the restriction endonuclease. Okay, this we have already seen in the previous class. Okay, and other than, if we are talking about the cloning, then we have talked about the plasmid, that it will going to work like the cloning vector. Okay, then other tool which we have talked about is nothing but a wait for a second. Okay, wait for a second. हेलो, कौन बोल रहा है? हाँ बोलिए। मैंने कल लगाया था, आज वो आया नहीं, सुबह 10 बजे का बोला था मैंने आने का। नहीं तो आना चाहिए था ना, मैंने सुबह कहा था, वो आए नहीं। अरे यार, तो फिर आपको हाँ या ना बोलना चाहिए। आपने तो कल रात को खाना बोल दिया था, फिर आज सुबह आना चाहिए था। करवाया नहीं, करवाना है। सुबह से हम बैठे हुए थे, मैं सोचा आएंगे शाम को कॉल करेंगे, अभी तक नहीं आया। हाँ ठीक है, आप किसी को भेज दो और करवा दो स्कूल में। देखो मैं जितना देना था मैंने उसे दे दिया था, बाकी मैं अभी बाहर हूँ। आप भिजवा दो, उस रूम में मिल जाएंगी मैम आपको, तो क्या कहते हैं, आप अभी साफ करवा दो उसको, फिर वो सब बाद में देख लेंगे। मैं थोड़ा बिजी हूँ, ठीक है, मैं बाद में बात करता हूँ। Okay, so what we are doing, we are talking about the tools. So the next tool comes the Fevicol, and Fevicol is nothing. This Fevicol is representing that we are talking about the any tool which will work as adhesive. Okay, which will going to work as the adhesive. Okay, adhesive which will going to join two different DNA strand. Okay, two different DNA strand. So you can see the name here, ligase. Okay, and as I have already told in the previous class, then whenever you will going to get these two words, means the ligase or lysis. So the ligase will means the breakdown. Okay, or to cut some sorry, not breakdown, what I am saying, ligase means the adding something. Okay, as we are using the ligase here na, so ligase means adding. Okay, and lysis means the breakdown. Okay, so the tool which we will going to use in the RDT, okay, RDT will be DNA ligase. One is DNA ligase, other is restriction endonuclease enzyme. Okay, and other than restriction endonuclease enzyme, what we are using? We are using the cloning vector. Okay, we are using the cloning vector, and other than the cloning vector, the last one is the competent host. Okay, competent host. So these are the four important tools which we're going to use in the recombinant DNA technology. Okay, this we have already seen in the previous class. So today we will only going to talk about the what? We will going to talk about the first tool of the biotechnology, that is called as the restriction endonuclease. Okay, we will going to talk about restriction endonuclease. So what I have told you, in the next lecture we're going to see the cloning vector and competent host. Today just we'll go through the restriction endonuclease. So first we will talk about REE, that is the restriction endonuclease. So in front of you are the three words, that is the restriction, endonuclease, and the enzyme. So let's first talk about the restriction. Whenever we talk about the word restriction, means we have some of the limits. Okay, we have some of the limits. So restriction endonuclease enzyme have property of cutting DNA strand, DNA strand in, sorry, on particular sequence. Okay, these restriction endonuclease enzymes are having a restricted property, or we can say property which limit their cutting power. Means they will not going to cut anywhere on the DNA. If there is DNA like this, okay, so they will not going to cut anywhere on the DNA. They have their own particular sequence, and we have a numerous number of the restriction endonuclease, not one, not two, up to 900, more than 900 restriction endonuclease we have discovered till now. So according to that, every restriction endonuclease, okay, every restriction endonuclease enzyme have its और ये लोग कोई पूछ भी नहीं रहे।
सर क्या कुछ पूछना आवाज आ रही है?
Anyone am I?
Yes.
Am I audible?
Yes, sir.
Yes, sir.
Okay, fine. Actually, there was something technical mistake, so due to that, I think so your screen display has stopped. So, and can anyone tell me where this screen is stopped, where I am on this topic? Can anyone say what I am
You said
telling?
about its property.
So I am here only, na? We are discussing this topic only, or the screen has gone before?
You started this topic, sir.
Okay, fine. So we started this topic. So we are talking about the restriction endonuclease enzyme. And as I have told you that why we are calling these enzymes as the restricted. Okay. So that is what a reason I'm telling you. So I will tell you reason once again. I think so because screen had stopped. So what we are doing, we are talking about the restriction endonuclease enzyme. So why we are calling it restricted? Because if assume this is one DNA molecule. 1 minute. Better make it good. Okay, so we are talking about that this is one of the DNA molecule, and what this restriction endonuclease enzyme is doing, it will going to cut the DNA. But what is the property of restriction endonuclease enzyme? That it will going to cut the DNA at specific sequence. Okay, it will going to cut the DNA of the specific sequence. Means we have the numerous number of the restriction endonuclease enzyme, not just one or two. Okay, so if we have the numerous number of restriction endonuclease enzyme, and each and every restriction endonuclease enzyme has its own specific sequence. Means each will going to cut at the particular specific sequence. And when we are talking about the specificity, means when we are saying that they are having something specific, so whenever we have the specific thing, so we are restricted to a particular property. Okay, when we have something non-specific, it can work on everything. Okay, but when the specificity will come, it will be restricted. So we can have an example that any restriction endonuclease enzyme, if get their specific sequence between this, so they will going to cut here. Or other than if they will, or another restriction endonuclease enzyme which is getting a particular sequence at this place, they will going to cut here. So the restriction has present here. Okay, not non-specificity, other than specificity is present. So this is all about why we are calling it restriction. Okay, why the restriction, what you are seeing here. Now we will going to start about what the restriction endonuclease enzyme has come into an existence. So in the 1963, as written here in the 1963. Now question comes, what happens in the 1963? So in the 1963, two enzymes, okay, two enzymes were discovered. Okay, two enzymes were discovered, and these two enzymes are discovered from whom? These enzymes were discovered from Escherichia coli, or you can say E. coli. Okay, because Now you have not to be concerned more with the names of these different organisms, but you have to know, because sometimes they will not give you the E. coli, or they will just give you the species that is Escherichia. So you have to remember that this E. coli, E means the Escherichia. Okay, Escherichia. So you have to remember that. Okay. So what this Escherichia coli have done? Two enzymes we will going to be discovered and will have discovered from the Escherichia coli, and how we have seen these enzymes as they are restricting. These enzymes are restricting growth of growth of bacteriophage. Bacteriophage. Okay, so what they are doing, they are restricting the growth of bacteriophage. And how they are restricting? As the bacteriophage means, if I say this is the E. coli, and as the bacteriophage enters inside the E. coli, these restricted enzymes which are present in the E. coli are breaking genetic material, genetic material of bacteriophage. Okay, if the genetic material of the particular organism has been break into pieces like this. Why into pieces? Because the restriction endonuclease enzyme cut the DNA at the particular places. So they will going to cut the DNA of the bacteriophage or the genetic material of the bacteriophage, and ultimately the genetic material will come into the fragment. So ultimately bacteriophage will not going to grow in the E. coli. So we have got these some of the enzymes, two enzymes from the E. coli. And scientists have thought to use this natural science in the recombinant DNA technology. When we have started biotechnology, I have already told you that what we are discussing in the biotech, we will use we will use natural science. Okay, we will use natural science. So what is the example of natural science here? These restricting enzymes. Okay, these restricting enzymes which are restricting growth of the bacteriophage in the E. coli, these are showing the natural science to us. When we are seeing the natural science, so in our mind what will going to come, that we can use these enzymes, we can use these enzymes. And then what we will use these enzymes, we will use in the RDT. For doing what? In RDT we can use this restriction enzyme as the molecular scissors. Okay, so this is the overview which I have all given you. Okay, so now we will move further. So now what we will going to talk about, we will going to talk about, as we have already seen, I am writing again, so I think so we will take the screenshot. In 1963, two enzymes were discovered from E. coli, which are restricting growth of bacteriophage. Okay, and I have already told you how they are restricting the growth of bacteriophage. Now, after that, we will going to talk about the nucleases. These are the white class of the restriction endonuclease. Means when we are talking about the restrictions enzyme, so restriction enzymes, okay, restriction enzymes are also called as the nucleases. Okay, and these nucleases, okay, these nucleases are two type. Okay, under the nucleases, two type of nucleases will come. If you have gone through the genetics, I know that you know the meaning of the nucleases. Can anyone tell what do you mean by the nucleases? Say soon. What do you mean by the nucleases? You
Sir,
have
it is an enzyme.
nucleases. Say enzyme.
That will break phosphodiester bond.
Good, exactly. They will going to break the phosphodiester bond. Ultimately, they will going to cut your genetic material. If we will talk on the overview, okay? Why? Because we are saying the biotechnology. We are not going through the genetics here, but we know what are nucleases. So nucleases are of two type: exonucleases and the endonucleases. Do anyone heard about the exonucleases and the endonucleases? If you will keep interacting and answering, you will going to understand better. Say soon. Okay, no. So first comes the exonucleases. So whenever the word is coming here, the exo means the outside. Okay, and we know endo means the inside. So exo is what coming here means is the outside. Means what is happening that this is your DNA molecule. Okay. So what will going to happen? The exonucleases when come into the working, these exonucleases will remove the nucleotide. Okay, they will going to remove the nucleotide from the ends. They will going to remove the nucleotide from the ends, okay, of the DNA. Okay, and when we are talking about the endonucleases, when we are talking about the endonucleases, here is what coming endo, which means the inside, endo under, exo bahar. Okay, so endo inside. So what will going to happen if this is the DNA molecule? Okay, if this is the DNA molecule, so what will going to happen? Endonucleases means the nucleases which will going to cut the DNA into fragments, but not at the end points. Okay, not at the end points. They will going to cut the DNA at the specific position within the DNA, within the DNA, like this. Okay, they will going to cut the DNA at the specific sequence, okay, within the DNA, not on the end. So that was the difference between the endonucleases and exonucleases. Okay, now we will go through what is the function of the endonucleases, because we need endonucleases, we don't need the exonucleases. Okay, because most of the desired gene which we want will going to be present within the DNA. Mostly they will not going to present at the ends. Okay, because at the end there will become the restriction genes, or the, we can say, telomerase point. So most surely we need the endonucleases. So we were going to talk about the functioning of endonucleases, that how endonuclease work. So each restriction endonuclease, okay, what they do, if this is your DNA, okay, if this is your DNA, so they will going to inspect, they will going to start the inspection. Okay, just assume that this is the restriction endonuclease enzyme. This yellow one is the restriction endonuclease enzyme, as I have already showed you here. Okay, like this. Okay, this is the what, this is restriction endonuclease. So what this restriction endonuclease will do, what it will be the functioning, they will going to inspect, means they will start the inspection. And inspection for what? I have already told you that endonucleases will going to cut the DNA within, okay, within the DNA they will going to cut the strand at the specific position. Means they are searching for the specific sequence. Okay, they are searching for the specific sequence, and whenever we search for, or what we do the inspection. And why the inspections are done? To see that everything is right or wrong. So why are the endonuclease is doing it? Endonuclease is doing it for searching the its specific sequence. So it will going to inspect, नहीं searching desired gene, so we will going to search that desired gene. Okay, first we will going to study about the desired gene. When we know that the desired gene have some particular sequence, then we will going to search the restriction into nucleus which have that a specific sequence or related to that specific sequence. Okay, then we will put that restriction endonuclease enzyme in on the working. So what this restriction endonuclease enzyme is doing, it is first start inspecting the length of DNA sequence. Okay, it will start the inspection of complete length of DNA sequence. And finally, what will going to happen after inspecting the DNA sequence? They will going to find specific sequence. Okay, their own specific sequence. And when they will going to find the specific sequence, they will going to cut. They will going to cut each strand. Okay, they will going to cut both of the strand, and as the strand is cut, we will surely, if the strand is cut, what we're going to get, we will going to get ultimately the our desired gene which we are cutting by that restriction into nucleases. Okay, so what we will going to get, we will going to get the DNA into the fragments. But so it is not so easy to get our desired gene. We have to do more working on that. First, what we have got, we have got the fragments. Okay, okay, so at the above, this is the original DNA, and now this is the DNA which is now cutted into fragments. Okay, distributed into the fragment. Just why? Because the restriction endonuclease enzyme is working on that particular DNA. Okay, so this is all about the restriction endonuclease enzyme. Let's go further and talk about the specific sequence, of which specific sequence we are talking about in the functioning of restriction endonuclease. Okay, so in front of you, it is written the palindromic nucleotide sequence. So this is nothing, this is that specific sequence, a specific sequence for restriction endonuclease enzyme. Means each and every restriction endonuclease enzyme which is discovered or which is present or exist on this planet will have their own palindromic nucleotide sequence, specific palindromic nucleotide sequence. Now the question comes, what do we mean by the palindromic? Okay, what do we mean by the palindromic? Okay, GATTC or G, 1 minute. GA. Can anyone tell me its complementary? We have all gone through the genetics. Tell me its complementary nucleotides. GATTC. What will be the complementary of C?
G.
So complementary of C is G. T,
A,
A,
again A,
T,
and
T,
T,
TT and
C.
C,
C. Now, so you have already, you have by own told me this nucleotide sequence. Okay, and if you will notice that these nucleotide sequence are same, are same, GATTC and GATTC, but when read in a single orientation, means if we have started from the 3 to 5, okay, and going to 3 to 5, when in the same orientation like this, GATTC, or if you will go in this orientation only, you will get the same sequence, GATTC and GATTC. These type of sequences are called as the palindromic sequences. Okay, again I am telling you This is one of the example, GATTC, and when you will form their complementary strand, so the sequence will come again the nucleotide GATTC, and when you will read this sequence in one orientation, okay, don't do like this, that GATTC and CTAAG. Now it is not right. Okay, why? This will not going to be called as palindrome. When you will read the sequence in one direction, okay, or in the one orientation, like we are doing. If you are going 3 to 5, then go 3 to 5 only. If you are going 5 to 3, then go 5 to 3 only. If you are saying from 5 to 3, CTTAAG, then you will get CTTAAG also at the other side. Means this type of sequences are the example of palindromic sequences. Okay, these are the example of the palindromic sequences. Now we will going to read the theory. So you have understood the concept. Let's go towards the solution. So what is the palindrome? Palindrome in DNA is the sequence. Is the sequence of what? Sequence of the base pair that reads on the two strands when orientation of reading, what orientation of reading is kept same. Okay, as we have already seen this, that when the orientation is kept same, 3 to 5, 3 to 5, 5 to 3, 5 to 3, then it will going to be same at the both side. So all the restriction endonuclease enzyme have the property, all the restriction endonuclease enzyme have the property of recognizing these palindromic sequences. Okay, and each and every restriction enzyme have its own specific palindromic sequence. Each and every restriction enzyme have its own specific palindromic sequence. Okay, it will going to have its own palindromic sequence. Okay, let's go and see further what it is written. So what the restriction enzyme will do? Restriction enzyme will cut the strand of DNA. Okay, one more property is coming in front of you of the restriction endonuclease enzyme. Understand it very carefully what they will going to do. Okay, what is exactly happening? If assume this is your DNA. Okay, if this is the DNA strand to say double helix. Okay, so what they will going to do, they will, if they are cutting the strand or they have searched or inspected the complete DNA sequence, and now within these two red lines they have got their palindromic sequence. So what will going to happen if the sequence is palindromic, GATTC and GAATTC. Okay, so this is the palindromic. But there comes the one of the property of whom? The restriction endonuclease enzyme. So what they will going to do? They will going to cut these both the strands just away from the center. Okay, let's go to the center, GAATTC. So according to it, center should come here. Okay, center should come at this place. But what they are doing, they are cutting both the helix of the DNA or both the strand of DNA just away from the center. So they will going to cut it from here and here. Okay, they will going to cut it from here and the at this place. Okay, just away from the center. This is one of the property of restriction endonuclease enzyme. You can see here EcoRI1, EcoRI. Okay, EcoRI is one of the name of the restriction endonuclease enzyme. After just telling you about the palindromic nucleotide sequence, I will going to tell you how the nomenclature of restriction endonuclease enzymes are done. So in front of you, these are the restriction endonuclease enzyme, and one of the example of restriction endonuclease enzyme is given here. That is the EcoRI. Okay, this is one of the restriction enzyme. So what they are doing, if you will going to notice that this is the palindromic sequence, so they are not cutting at the center or they are not cutting at this place. What they are doing, they are just cutting this strand just away from the center. You can see here, they are just cutting away from the center. See this. Okay, so ultimately what will going to happen? Sticky end will going to form. Why we are calling that sticky end will going to form? Just tell me if this is the DNA or we are saying that this is the double stranded DNA. Okay, double stranded DNA means both of the both of the strand strand contains strand contains complementary base pairs. Complementary base pairs. Okay, and if they are complementary, so what will going to happen? They have the tendency, if something is complementary to each other, if I am saying that this is the block, okay, this is something block, and you have socket like this. Oh, sorry, what? You have something box like this, and you have something socket like this. Okay, so this box, okay, this box have a complementary size with this particular socket. Okay, so it will going to adjust in this socket very well. Same like this same, what will going to it will going to happen with this DNA strand. What will going to happen that if these both strands are complementary, that is what a reason we are calling them double stranded DNA. So both have the complementary base pair. So if they are complementary, they have the tendency to attach with each other. Okay, they have the tendency to attach with each other. And what is happening if you will go through this? Wait, I will make you understand whole. So understand till now that tendency to attach with each other they have. Why? Because they are containing the complementary base pair. Now remove everything. I will make you understand from the beginning. So, how we are using the restriction endonuclease enzyme? We are taking the DNA. Okay, we are taking two different DNA. One DNA belongs to the uh, that one DNA belongs to the plasmid. Okay, this is the plasmid DNA. Okay, plasmid DNA means we are talking about complete plasmid. Plasmid is a circular small double-stranded DNA. Okay, so this is the plasmid DNA, and what this is? This is the DNA which is containing the desired gene. Oh, 1 minute, wait. Okay, so we have the two DNA strands. So both the DNA strand, one belongs to the vector DNA. Vector DNA means when we will use them as the vector end. Other is the foreign DNA. Okay, which is coming from the outside and we are using means it contains the one of their desired genes. So what we are doing, we are using from side the vector DNA which will going to carry that particular DNA or the particular foreign gene of foreign DNA. So what we are doing, we are cutting both these DNA, both these DNA with same restriction enzyme, with same restriction enzyme. Okay, and when we are cutting both the DNAs with the same restriction enzyme, so they will going to cut both the DNA at the same positions. Okay, so when they will going to cut different DNA at the same position, now what is happening? See here. See here and see here. These restriction enzymes are same and they are cutting both the DNA at the same position. So the sticky ends will form. Why? Because the complementary base pairs are left. If you will go through this, so what is remaining here? TTAA, and the other side on the foreign DNA, what has remained? AATT, means these both are complementary to each other. And if they are complementary, they have the tendency to attach with each other. That is what the reason we are calling these ends of the DNA as the sticky end. Okay, and when we have the sticky end, so what we will going to do? They will going to attach with each other. Other than these sticky ends, okay, these sticky ends will will facilitate working of whom? Working of DNA ligase. Because what is the work of DNA ligase? DNA ligase is working like a favicol, means it is attaching two different DNA. Okay, that is this work. Okay, this work of attaching two different DNA is of the DNA ligase. But having sticky ends or the complementary base pair will help, okay, will work as the additional help to the DNA ligase to attach these both of the DNA. So this is the working of the restriction endonuclease enzyme. Okay, now I have to tell you some more things about this restriction endonuclease enzyme. When you will go through the NCRT, you will going to read about the some of the history of the restriction endonuclease enzyme. That there is the first restriction endonuclease enzyme called as the Hind second. Okay, so you can read that all theory by book only. Okay, wait a minute. Now, what we have to see that nomenclature of restriction enzyme. Okay, now we will going to see how the nomenclature, as we have discussed one name, na EcoRI. So as the same, we will going to discuss how this nomenclature or the names given to a particular restriction enzyme. Okay, so the according to the conventional naming, means the naming which we are doing from the very previous time, we are using that method only. So what we are doing, we are using the name and we are forming the nomenclature as first letter of any name of the restriction enzyme belongs to its genus. Okay, then the second two letters belongs to the species, means the first letter will come from the genus from which we are getting this restriction enzyme. I am telling you again that the first letter of the name of the particular restriction enzyme will come from that particular organism, genus of that particular organism from which we are getting this restriction enzyme. Okay, as we have already discussed now, E. coli, that from the E. coli we are getting the two enzymes in the 1963. So according to that, the genus of the E. coli will going to be the first letter of the particular restriction enzyme. So the first letter belongs to genus, second two letters belong to the species, okay, and other letters, okay, other letters will belong to the order in which they are discovered. Okay, means if you are seeing the any Roman numbers, if you are seeing any Roman numbers, so these Roman numbers are nothing but they are indicating the order, indicating order in which any restriction endonuclease enzyme are isolated. Okay, just any particular enzymes are isolated from any particular organism or any species genus. So we know as from the E. coli we are getting the two different enzymes. Okay, two different restriction enzyme we are getting from the E. coli. So the Roman number can come in front of those restriction enzyme as the restriction enzyme 1 or restriction enzyme 2. So these Roman number 1 and 2 are representing nothing, but they are representing in which order these are isolated. If any particular enzymes are isolated first, so the Roman number will come the 1. The restriction enzyme which will come after that will going to be said as the 2. Okay, so this is the one of the type of the nomenclature we will use for the restriction endonuclease enzyme. And other than this, what will going to happen that in the example, okay, just see the example. So what is the example? Example is E co R I. Okay, this example is in front of you already. We have seen this here. Okay, that it is working as the restriction enzyme. So how this name has given E co R I? So E co, this comes from the Escherichia coli. Escherichia coli. Okay, now what we have told you that after the Escherichia coli, what we will going to see, we will go towards the species. Okay, so the species Escherichia coli have the different species. So RY, this RY. Okay, different names are given at the level of the laboratory. So Escherichia coli RY and 13. So this is the name of the organism or name of the particular species, okay, from which we are getting this restriction endonuclease enzyme EcoRI. So eco, okay, what we have discussed that the first letter come from the genus, so genus is Escherichia, and the two letters come from the species, so species is what? Species is coli. So we are taking the two letters C and O. Okay, so it's eco, and RI is coming from the RY is nothing but the strain of the Escherichia coli. As I have told you during the human health and disease, as we are fighting with a particular pandemic, and that pandemic is due to coronavirus, but at present we have different 11 strains of the coronavirus, means small differences between the each and every strain of the coronavirus. But yes, 11 strains are present. The same type, Escherichia coli also have the different strains. So RY is the name of that strain. Okay, so from strain we are getting one letter R and one that belongs to the what? That belongs to the order in which the restriction enzyme are isolated. So eco R1 means we are getting this particular restriction enzyme first from the Escherichia coli. So this is the way of doing the nomenclature of any particular restriction enzyme which we are getting from any particular organism. Okay, so till now we have a numerous number of restriction enzyme around the, as in your NCRT there was a written data of the 900 restriction endonuclease, but that data is coming from very previous time. We are following that we have more than 900 restriction endonuclease enzyme now. We will go through different research. Okay, so as I've told you, if we have taken data of the 900, so we can think that 900 restriction endonuclease enzyme have different 900 palindromic sequence. Okay, so that is what I want to make you understand, that DNA or the genetic material or these different types of enzymes are not so simple. Okay, they are working on a very complex level. Okay, they are working on very complex level. You can think that 900 different restriction enzyme have different 900 palindromic sequence. So according to that, what we can say that yes, the variation can be possible or diversity is possible. Okay, so this is all about the restriction endonuclease enzyme. This much you have to understand today from the previous class. Okay, from the previous class we will continuing this topic, or I think so I have got the news that you all have to understand about the genetics. Is that right? You want to understand genetics once again? Aryan, am I audible?
यस सर.
जेनेटिक्स पढ़ने के लिए बोला था क्या?
नो सर, मैंने तो नहीं बोला.
नहीं, बाकी नहीं, ऐसा नहीं पूछ रहा हूँ, मैं सच में पूछ रहा हूँ क्योंकि मुझे बोला गया है जेनेटिक्स के लिए, सो आई एम आस्किंग कि जेनेटिक्स पढ़ने के लिए आप लोगों ने बोला था कि ऐसा ना हो कि बेवजह क्लासेस लगें, टाइम
नहीं
वेस्ट हो।
सर, नहीं सर, मतलब हम लोगों ने तो नहीं बोला. जिसने भी बोला वो तो शिवेन सर ने भी पूछ रहे थे कि तुम लोगों ने बोला है क्या, तो
हम्म।
क्लास ले लेंगे वो. मगर फिर ग्रुप पे भी डाला उन्होंने, मगर किसी ने कोई रिस्पॉन्स ही नहीं दिया.
हाँ तो इफ द टीचर इज आस्किंग अबाउट एनीथिंग दैट प्लीज आंसर इट, ओके? इफ यू आर नॉट डूइंग सो दैट इज अल्टीमेटली द disrespect of particular teacher, okay? So don't do these type of things. If teacher is asking, answer
चलो बे, अब नहीं आ रहे सर।
सर ने बोला।
अरे बोला तो नहीं है, अब चलेगा, टाइम भी हो गया, कैसे आएंगे?
पूछ लो यार एक बार, ऐसा ना
नहीं,
ऑनलाइन भी नहीं है।
व्हाट्सएप पे बात कर लो।
ऑनलाइन नहीं है
ना, ये गड़बड़ आई है पूरी मीटिंग में। अब चैट किसी को करना है, किसी को जा रहा है, क्या पता क्या है।
एक काम करो, सर को फोन लगा लो, राजुल सर को फोन लगा लो।
ऑप्शन इसलिए लिए हैं कि कहीं की चैट कहीं बजा, हद ही हो गई। देखो कॉल कर रहा है।
कॉल कर लो। दूसरी 10th वाले बच्चे भी ज्वाइन कर रहे हैं क्या? खत्म, चलो जा, आ गया ट्रैक।
क्या पता यार, वॉइस क्लियर नहीं आ रहे हैं शायद।